Good afternoon, everyone, and welcome to the Agro March webinar. My name is John Clark, and I'm here at the University, the snowy University of Massachusetts in Amherst, and I'm going to be the moderator of the webinar today. And today I'm extremely pleased to introduce Professor Jeff Bloomquist, who's at the University of Florida in Gainesville as today's speaker, and I'm just going to give a little bio here to make sure everybody knows who Jeff is if they don't already. Jeff, one of the little factoids in the world is he was born and raised in northern Indiana, which is interesting because after becoming toxicologists, we both realized that we grew up about 15 miles away from each other. And of course, we didn't know each other until we became sort of professional insecticide toxicologists. Anyway, Jeff received his BS degree from Purdue University in 1978, where he met Bob Hollingsworth, and a lot of you, I'm sure, know Bob, and kind of set Jeff on his path to become an insecticide toxicologist. And he then went on to Mississippi State University and received his MS degree there in 1981, and he worked with Dan Strickland. And then finally, he moved on to University of California, Riverside, where he got his PhD with Tom Miller in 1984. From there, he went off to Cornell University and did a postdoc with David Soderlin, and then finally took a position with Rome Planck in 1988. And I believe this is probably where Jeff really became interested in alternative sort of a new and novel chemistry for insecticides. From there, he took a position at Virginia Tech in the Department of Entomology in 1989, and then was promoted to a full professor in 2003. During that time, Jeff has developed an internationally recognized program in neurotoxicology, insecticide resistance, and of course, the search for new insecticidal molecules. At that time at Virginia Tech, he also worked on a number of projects, one of which was looking at the effect of neurotoxic insecticides in terms of Parkinsonism, and established a linkage between these kind of compounds and not only Parkinsonism but also diabetes, and so this was some of the first work where we start seeing some of these other effects besides just neurotoxicology. Jeff now currently leads a large program that's looking at the development of new compounds to control malaria mosquitoes. And this project was put together with one of the first Gates grants that was awarded for this process. And in 2009, Jeff then moved on to University of Florida, where he joined the Emerging Pathogens Institute. And in 2017, he was the recipient of the International Award for Research in Agrochemicals that's sponsored by the Agrochemical Division of American Chemical Society and DuPont Crop Protection. And so last August, when Jeff had his award symposium, he presented this presentation that he's going to be giving today that's entitled Mechanisms of Synergism for Increased Insecticidal Action. And with that, I'd like to introduce our today's speaker, Jeff Bloomquist.
Thank you, John, for that detailed and really effusive introduction. And welcome to everyone who's signed in to the webinar from sunny Florida. It's a little chilly today, only about 68. But we'll get through this terrible weather as best we can, John. In any event, I'd like to again thank the Agro Division and ACS for the opportunity to speak to you today, and as well as the sponsorship of the International Award, as John said, by DuPont Crop Science. I became interested in this idea of synergism basically from a perusal of industrial patents. We keep an eye out for these as we are looking for new chemistry. And we noticed that there was a growing pattern of these kinds of applications. Here's one from Valent Biosciences, where they were using a mixture of different kinds of fatty acids with pyrethroid adulticides, and claiming that they were able to overcome certain kinds of pyrethroid resistance mechanisms with these kind of mixtures. The example here in the middle has kind of an innocuous sounding title, pyrethroid insecticide for protecting plants and seed. But if you actually download this patent and look at it from BASF, it has a whole slew of different kind of mixtures with synergistic effects for these kind of agrochemical or crop applications. And the one down here in the bottom was interesting to us as well. This is from a DuPont patent, actually, going back to 2012, so it's a little bit old. But the title is Synergistic Mixtures for Controlling Invertebrate Pests Containing an Anthraniliamide Compound and Flonicamid. And excuse me, as I'll say later in the presentation, we were studying flonicamid as part of one of our new mosquiticide projects that we had from the foundation of the NIH. So to provide you with kind of an overview of synergism in its most simple-minded definition, we're thinking about the combined response of two compounds where each alone, when you add them together, it gives you a greater than additive effect. Maybe nothing more complicated than that. Synergism has been used in the field actually for commercial pest control and crop protection applications. Synergists are commonly employed in household products. It's also used to document, define, and overcome resistance in the field in some cases. And there are a number of different mechanisms of synergism, and the first of these that I'd like to discuss with you would be a metabolic blockade, which is really the most common form of resistance and the most common form of a mechanism of action of different kinds of chemical synergists. Among these are piperonyl butoxide, DEF, which is an abbreviation for defoliant. It's actually an organophosphorus compound, and diethyl maleate. So the usual way these are employed is rather simple-minded. We'll take piperonyl butoxide, co-apply or pre-apply it before an insecticide. If we see no effect, the conclusion is that there are no involvement or no significant involvement of cytochrome P450 monooxygenases in the action of that chemical. If we see synergism, okay, that implies a detoxification by P450s. And there are not a small number of instances where you can actually see antagonism, where you get bioactivation of a molecule by specific oxygenation steps catalyzed by P450s. In the center here, we've got DEF. The same kind of logic applies. No effect of the synergist means esterases are not involved in the metabolism of the molecule. Synergism tells you there's some detoxification by esterases. Antagonism, that there's activation by carboxyl esterases. With DEM, there are instances of synergism. These molecules, they will block detoxification by glutathione S-transferases. Obviously, if there's no effect, they're not involved. I don't know of any documented cases where GST type enzyme systems are responsible for biological activation. But there are issues, as it says here, with metabolic synergists. We apply these compounds typically at or near the maximally tolerated dose, something that's just sublethal, if you will, and we kind of blithely assume that they only have one physiological effect, okay? Synergists themselves can be toxic. You can overdose an insect with a synergist. That action is probably not related to its inhibition of metabolic enzymes. We assume that there's no crosstalk between enzymes and inhibitors. We know this is not the case. It's been shown now that piperonyl butoxide can block carboxyl esterases. It's not as good an inhibitor of carboxyl esterases as it is P450 monooxygenases, but they do have some effect on those kinds of enzymes. There's also a phenomenon that was documented back in 1972 in a really seminal paper by Sun and Johnson called quasi-synergism. And they showed that certain synergists like PBO can have effects on insecticide penetration as well as just on metabolism. And just to throw you another curve, there is a very interesting paper that was published a while back showing that both piperonyl butoxide and DEF actually slow the penetration of propoxer in the German cockroach. And we've occasionally seen where a certain kind of molecules will slow the knockdown action, at least, of the pyrethroids, even if they were to synergize, 24-hour lethality. Another mechanism of synergism, moving on from metabolism, would be enhanced penetration. There are these ABC transporters, as they are so named. They are able and commonly sit in the cuticular epithelium in insects. If you consider this a cell layer beneath the cuticle or the integument, the substrate, in this case the insecticide, will passively penetrate, grabbed by one of these transport molecules with the help of the energy of ATP. It will be extruded back out of the body, back into the pericellular spaces and underneath the cuticle. This delaying action of the penetration then helps to protect the insect from the toxicity of these molecules. It is not generally as effective a resistance mechanism as metabolism, but it is there, it is measurable, and there have been shown to be upregulation of these transporter proteins in some resistant strains of insects. There are certain kinds of molecules that are able to interact with these transporters. That's shown over here. This is a paper taken from the journal Parasitology. This is a larval feeding inhibition curve for Haemonchus contortus, which is a internal parasite, a roundworm, and it carries, in this case, an ivermectin resistant isolate, this MHCO4. And so if you follow the curve here, if we are looking at the percent of feeding, it drops down to about 60%. So 40% protection is available from ivermectin up to about 2 nanograms per mil. If you co-incubate the worms with pluronic 85 at 22 micromolar, valspadar at 5, verapamil at 50, or the fungicide ketoconazole at 10 micromolar, you can see they're all pretty effective at blocking this transporter and increasing the biological activity of ivermectin, shifting this curve down to you're getting basically complete feeding inhibition at 0.5 nanograms per mil. So this is another possible mechanism of synergism. You don't see it specifically deployed much, but I think that maybe we will more in the future. Another mechanism of synergism that I've studied in the past has been 2 agents acting on the same target site protein. For this particular example, we'll consider this diagrammatic figure up here on the right of the voltage sensitive sodium channel. The alpha subunit of this ion channel has 4 homology domains. It carries an almost dizzying number of different toxin binding sites. This particular iteration of the review, site 1 through site 7. For the purposes of our discussion, we're going to consider site 2, okay, in yellow, binding batrachotoxin or veratridine, and site 7 with the pyrethroids. Lacey Jensen and Troy Anderson have some unpublished data, which I think is very provocative though. And this is the LD50 concentration of topically applied compounds to the malaria mosquito Aedes aegypti. And so this is veratrine, which is basically a mixture of different alkaloids. Veratridine is what the veratrine class. And so this is the LD50 value you get for veratrine alone. When you apply veratrine with an LD25 of permethrin, you can see you get a very significant drop in the LD50 value of veratrine, much more than you would expect from additivity. And then with deltamethrin, the dose is becoming extremely low. I don't know what the synergist ratios are for these, but I'm sure this one looks like it's about at least, I would say, 6 or 7. This one is at least 10. So very strong synergism by these 2 classes of neurotoxins acting at different binding domains on the same target protein. I've been involved in a number of years with a similar but complementary kind of an idea of 2 agents acting on the same target site protein, and these are bivalent anticholinesterases. I worked on this with Paul Carlier, a collaborator of mine for near 15 years now. And these are molecules that, in this case, this end of this warhead, if you will, binds at acetylcholinesterase near the catalytic serine, here labeled 199. All right, so this is the carbamate moiety binding in the acyl pocket of this enzyme. This is the pyrazole group forming a hydrogen bond with this tyrosine. And then we have this carbon link tether of 3 carbon, this alkyl chain. And it then allows this other moiety on the phthalimide to hydrogen bond to what's known colloquially as the peripheral site. So this peripheral site sits at the mouth of a catalytic gorge. And when the substrate comes down here, it is passed to the catalytic site. This molecule is able to bind to both of those sites simultaneously. And it's occupied together and linked, forced propinquity with that alkyl chain. We get a pretty reasonable topical LD50. G3 is a susceptible strain of Anopheles gambiae, the malaria mosquito. The LD50 of acron, which is highly resistant to, essentially insensitive to molecules like propoxer. We get 386. The resistance ratio here is only 6. Whereas it would be several hundred-fold if you could measure it all with propoxer. And if we did this experiment by injection in susceptible, we get a number at a fraction of a nanogram. So the binding capability of this molecule when it reaches the hemocoel is really pretty good. So it's able to get through the blood-brain barrier as well. The cuticle, though, is a bit of a barrier to it getting in there. I'd like to move on now and talk about physiological synergism of two agents acting on different target proteins. And there are lots of examples of this. I've picked this one from Vince Salgado's paper that he published back in 1992. He was one of the first scientists to show that this class of compound from Roman Haas were able to block voltage-sensitive potassium channels. And we were very interested in those. And he combined these molecules with fenvalerate, the pyrethroid. And he did something that for synergism is called an isobologram plot, and that's what's shown over here on the left. So basically, this curve or this point at the origin, 0 dose of RH1266, this is the LD50, if you will, of fenvalerate. And then there's another point out here, okay, this is with no fenvalerate, this is the LD50 of RH1266. This point M is a combination of about 6 micrograms of one and about 3 nanograms of the other. And so you end up with a point down here, and anything that falls in this region of the curve is actually below this line is indicative of synergism. And if you look at the distance between O and N, the origin and N, this curve, where it bisects this line of the two LD50s, and this is the data point for the combination. The ratio of those the distance is about 3, and that's indicative of the synergism ratio that you are getting when you combine these two molecules together. So this is the kind of analysis that was done for synergism for a number of years. It's also for us was an indicator that potassium channel directed compounds might be useful synergism for pyrethroids. We had a project that was funded by the NIH to look at new compounds affecting this class of target. And I'd like to spend just a minute reviewing potassium channels so that we're all on the same page with regards to the neurophysiology. So this is a very famous plot actually taken from a figure published by Toshio Narahashi. And this is a typical action potential waveform. You have a depolarizing upstroke, and this is due to a rapid increase in GNA is the conductance of the nerve membrane to sodium. Sodium flows in, the membrane becomes more positive, this conductance shuts off, you have a delayed then potassium conductance that activates, potassium flows out, brings the membrane potential back down to resting level. Now, there are two main classes of potassium channels in axons and other kinds of excitable tissues. Certainly, these are not the only two. There are many classes of potassium channels with many different kinds of properties. But the I sub K, this type of current, also called a delayed rectifier, that is responsible for this part of the potassium conductance in a squid axon or a cockroach axon. There's another kind, though, called the I sub A. This is a rapidly activating and then inactivating kind of a channel. So it passes potassium, but it has different voltage-dependent kinetics than what you see in I sub K. And a newer nomenclature for these is, KV1 and the KV2, basically those two kinds of families of voltage-activated potassium currents. So we've been looking at potassium channel blockers. There are natural and synthetic ligands that exist for these channels, so we had the opportunity to look at different kinds of lead compounds. We reasoned that they should target both nerve and muscle. They should not have to penetrate into the CNS, which is a problem that we ran into with our cholinesterase inhibitors, like that bivalent molecule I just showed you. It's pretty active, but it has problems going through barriers, in that case, the cuticle. So if we can work in the periphery, that would be an advantage. We thought these compounds would be lethal and perhaps also excitorepellent, which is something you see with the pyrethroids. It's a novel site of action, so we should have low cross resistance. And through cheminformatic analysis by my colleague Max Totrov, who works at Molsoft in San Diego, a commercial molecular modeling firm, he found this molecule, which is also published in Nature Chemical Biology back in 2012. It's a substituted benzamide with this somewhat unusual cyclopentyl group. And this is called 2S65-465, and we found that it had some activity against Anopheles gambiae, not great, but some. Its oral mammalian toxicity, though, was very low. It killed 0 of 3 mice that we treated orally at 2,000 milligrams per kilogram. But if you compare this to, Anopheles gambiae toxicity of propoxur, it's about 200-fold less active. But this compound is much more toxic. It's 100 milligrams per kilogram in the rat. Permethrin, again, another order of magnitude and change, more active than propoxur, with low rat toxicity as well. So we studied these compounds on Aedes larval muscle. So we were impaling the muscle with recording microelectrodes just to look at the membrane potential and any kind of spiking activity that might be happening from a presynaptic effect. So we have the motor nerve that's innervating the muscle, and we're recording how that nerve is talking to the muscle and how the muscle membrane potential is being maintained. And so we get a nice resting potential in these muscles, and we put on increasing concentrations of 2S. And at 10 to the minus 5th molar, we started getting these volleys of EPSPs. Probably indicative of the fact that the presynaptic nerve terminal is being poisoned by this compound and is eliciting spontaneously, without being stimulated electrically, these long trains of EPSPs. We also tested this compound against, strangely, headless mosquito larvae. We developed an assay that helped us to evaluate the intrinsic toxicity of molecules that might not penetrate the insect integument very well. And so we found that if you pull the heads off of mosquito larvae, they'll live quite well for about 5 hours and remain pretty responsive to probing. So that if you soak these animals in a treatment solution, it now has entrance here in the cervical region, a diffusion pathway into the body itself, and then is able to exert its effects. And 2S causes this twisted, contracted kind of an effect on the whole mosquito that's pretty much reminiscent of what we see when we look at the physiology of the skeletal musculature. In addition to this, we had engineered a HEC-293 cell line to express the Anopheles gambiae Kv2.1 channels, and we evaluated those under patch clamp conditions. So this is, if you will, a typical kind of a HEC cell. Here's our patch electrode, which is able to maintain membrane potential and look at the resulting currents. And this is just a percent of control current being inhibited by 2S, and it has an IC50 value of 166 nanomolar, which is a pretty good value. We've done the same kind of an experiment on a human neuroblastoma cell line, SY5Y, that will express the Kv2 channel from the human. We get a nice inhibition curve. The IC50 value, however, is not much different than what we see in the mosquitoes, so there's not much selectivity between the mosquito channel and the human channel, unfortunately. My PhD student, Xiao Zheng, presented this data at the same meeting back in Washington last August. He's been working on the whole mosquito synergistic interactions of the 2S65-465 potassium channel blocker, and in this case, looking at its effect on the dose response lines that we get for Anopheles gambiae strains in whole mosquitoes. Okay, so the G3 permethrin response line is shown here in the filled circles. Okay, so this is the response line for the susceptible with permethrin alone. This line over here is the acron. It's a pyrethroid resistant cell line, so the curve is shifted to the right. This is acron with 2S, okay? So the resistant line moves over and they become basically susceptible, the same as a susceptible LD50. And the susceptible line, though, of course, jumps quite a lot when you treat the susceptible strain with 2S, a fixed dose of 125 nanograms. We get about 7-fold synergism at the LD50 level. That's the interpolated line here at 5 probits, drop down then to calculate LD50s from the x-axis. So we get about a 7-fold shift in the susceptible strain in the permethrin dose response curve. We get a 3-fold shift in the resistant strain. We've done the same experiment. Remember, this is a KV2 channel blocker. For pyridine is also a channel blocker, but it's the KV1 type I sub A current, if you will. It only gives us about 2-fold synergism. So 2S is a much more effective synergist for permethrin than for AP. And we've compared this with PBO synergism as well. And Xiao has found that co-application with PBO gives us about 2-fold synergism, or we get 4-fold synergism if we pre-apply the PBO, wait 4 hours, and then apply the permethrin, which is generally a little bit more effective. It gives more time for the synergist to penetrate and interact with metabolic enzymes. But in any event, any of these permutations of treatment, 2S is the best synergist so far that we have seen from permethrin against the malaria mosquito. And we're pretty interested and excited about learning more about the mechanism that's underlying this effect. To give you a pictorial representation of what happens to the mosquitoes, Xiao has made some videos here. This is an LD10 dose of 2S. And one of the things that we see with some chemicals is that they have leg drop. When mosquitoes are poisoned, their fine, wispy legs will be autotomized, as it's described, probably from disruption of the neuromuscular system. And we have a video of this. It's not going to let me do it unless I go back to my arrow. There we go. Okay, so these are LD10 doses of 2S poisoning adult Anopheles gambiae mosquitoes. You can see they have disrupted flight. Their posture is all wrong. They're definitely intoxicated. There are some bent legs here, but they're not really on their backs. It's rather a mild poisoning syndrome at this dose. This is the LD50 of permethrin. You can see we have more intense poisoning. The mosquitoes, many of them are laying on their sides. A lot of convulsions, convulsive flight when they're disturbed, right? The dropping of the cup. And the other thing you see here are the legs have dropped. There's leg drop here. Not a lot of them, but you can see here's 2 legs, 4 altogether. There's another one over here. And so we get some leg drop at the LD50, no leg drop with 2S. But when we combine them together, if you look at the picture here on the right, you can see there's legs laying all over the place, okay? They shed their legs very readily. And if you watch them very carefully, especially this one here and some of the others, when they fall off, the legs are moving. Okay, see it's moving over here. The legs are undergoing spontaneous contraction because I think of the combined effects of potassium channel block and the pyrethroid effect on the sodium channels. And so you get a little bit different kind of a poisoning syndrome when you combine these two together. I'd like to move on now and talk about flonicamid. I mentioned when we were looking at the patent applications, the one from DuPont talking about flonicamid and some of the diamide compounds that are ryanodine receptor activators. This compound, flonicamid, has a novel behavioral action. It's an aphid antifeedant. It's not a frank neurotoxicant, okay? It is very amenable to chemical substitution. It's a pretty simple molecule. It has a low molecular weight. It's pretty simple. Maybe it reminds us of DEET a little bit, maybe a little bit of 4-aminopyridine. The other thing I would like to point out is that it's been shown that flonicamid itself is not the actual toxicant. It's a pro-insecticide. This primary amide, TFNA-AM, trifluoromethyl nicotinamide, or sometimes we refer to it as PRC 1369, as it was resynthesized by Paul Carlier, is this molecule. And so we've studied flonicamid, the parent molecule, as well as its primary amide metabolite. And we find that flonicamid is an antifeedant in mosquitoes, at least it looks that way, but we don't get a lot of activity. This is a sugar feeding assay conducted like this, mosquitoes in a test tube with a cotton stopper with flonicamid laced sugar water for sustenance. And if we monitor the mosquitoes over 7 days, the control, we get a little or no mortality. Okay, and if we treat with either 1369, which is the metabolite, or flonicamid itself, but it's a high concentration, 1,000 ppm, we're able to kill about 30% of the mosquitoes at 25 to 30% at best. It's a statistically significant effect, but it's really not a very effective insecticide this way, and it's not marketed against most insects, just aphids. It's very effective against aphids as opposed to other types of sucking pests. So this wasn't too much of a surprise. Early mode of action studies, and this actually goes back to a meeting report by John Hayashi and his colleagues at FMC, who licensed this material, and we're studying it back in 2007. These are patch clamped insect neurons from Heliothus virescens, okay? So it's a lepidopteran, and you have two kinds of currents here. There's that early A current and the late I sub K, the delayed rectifier current, and this is the control in saline, and then saline plus flonicamid. You can see that both the early inward current is suppressed as well as the late persistent current. So it's blocking both types of potassium channels, at least in these kind of neurons. Does not have any action on nicotinic acetylcholine receptors. I think because of its chemical structure, it was called flonicamid, and it's a nicotinamide, but that kind of name doesn't have anything to do with its target. So we've studied these molecules. Again, we're looking at Aedes aegypti larval muscle and comparing them in this case to 4-AP, because it's more of a structural analog to flonicamid than 2S would be. And here's the 4-AP response. You see at 1 micromolar, and then at 10 micromolar you get some repetitive firing induced in the nerve, and then a very strong depolarization of the muscle membrane potential. Flonicamid up to 1 millimolar, it starts to have a small effect at 1 millimolar. Anything beneath that, we never saw any effect. So it's a rather weak inhibitor of potassium channels. PRC 1369, or the amide metabolite, is a little bit more active. We start to see a small effect at 100 micromolar, and then 1 millimolar here. We went ahead and patch clamped our HEC 293 cells. These are expressing the KV2 channel, not the KV1 channel. It's the primary target site of 4-AP, and we get basically overlapping concentration response curves with a threshold of about 100 micromolar and about 50% inhibition at 1 millimolar. So they're not very active on potassium currents or on tissues that would be reflective of a potassium channel blocking effect. In terms of toxicity, okay, we've treated flonicamid and the metabolite on Aedes aegypti. This is our susceptible Orlando strain. And if you look at 1 hour knockdown, it takes this kind of a dose to give you 50% knockdown, about 10-fold more active though if you apply 1369. It's 11-fold, so it's a much more respectable kind of a dose. A 24-hour mortality though with both compounds is greater than 10,000. We never see more than about 10% mortality here. So there is a knockdown effect, but not much lethality. In Anopheles gambiae, we get about the same KD50s values for each. However, at 24 hours, we do see increased knockdown, lethality. The LD50s go from more than 10 micrograms per mosquito down to about half a microgram, an increase of at least 20-fold. And then with Anopheles gambiae, down to 38, so a 263-fold increase. So we do see here that Anopheles gambiae is much more sensitive than Aedes, and you tend to see this across different kinds of molecules. In particular, I think what this is reflective of is perhaps the ability of these compounds to penetrate through the cuticle of Anopheles gambiae maybe a little bit better than we do in Aedes aegypti. And here are some of the signs of intoxication. Again, by 10 nanograms of 1369. And especially if you look at these mosquitoes that have landed on the undersurface of the netting here, you can see that they have a very strong effect on the postural, loss of postural control in the legs. You get these extended legs and curled tarsi. And in discussions I had with Ralph Nowlin, who works at Bayer, he sent me these pictures, and these are from Ralph actually, showing the effect on caterpillars. This is published data from another paper, and then this is flonicamid, and then the flonicamid metabolite. And you can see that in the grasshopper, you get this two pronounced effects on posture. One is the grasshopper does not hold itself up anymore. It's down on the substrate, and the hind legs are lifted and extended like this. Very unusual sign of intoxication. And here's a close-up picture of one of our mosquitoes treated with the flonicamid metabolite. And you see we're seeing a very similar kind of a behavioral effect where the mosquito is down, it's not maintaining posture, and the legs are extended. Vince Salgado had published in a meeting abstract back in 2014 at the International Congress of Pesticide Chemistry that flonicamid was affecting chordotonal organs. Vince, I don't think that data has been published yet. We were interested in confirming if that was kind of thing that we would see in dipterans. And so we looked at dipteran larvae. This is actually a picture of a Drosophila melanogaster third instar larva. And if you look at the body wall under the right kind of morphological preparations and microscopy, you can see there are groups of these chordotonal organs, sensory organs that are in the body wall that report on the position of the different segmental arrangements in the larva. Okay. We've gone ahead, because of the high conservation that you see in these kind of neural circuits across insect orders. We have not confirmed with microscopy that these exist in housefly, but it's a lot bigger larva to work on than Drosophila. And so we generated a preparation that had been published in the literature before, looking at a eviscerated third instar housefly larva, then recording from these peripheral nerves with a suction electrode, using a small audio speaker, actually with nothing more than a glass capillary glued into the middle of it. But if you apply a square pulse to this, it moves this capillary a small amount, and if you push it up against the body wall, you can get both onset and offset nerve discharges from the sensory fibers that are in this body wall. And so if you look at a DMSO control, and this is doing a signal average of 20 sweeps, so it removes all of the spontaneous activity from other kinds of sense organs, and certainly there's more than just these chordotonal organs and these peripheral nerves. But you get then a response that's very well matched with the onset and offset of the pulse. And if you do put on 100 micromolar 1369, admittedly a very high concentration, but we're also showing this is a 100% effect. These effects, these little oscillations, that's actually just oscillations, mechanical oscillations from the onset and offset of this pulse, of this stimulus, this mechanical stimulus. And so these are not nerve related. These are just related to the mechanical system that we have to stimulate the body wall. Over here in the lower right is shown a concentration response curve for 1369. It has an IC50 of about 2 micromolar. Okay, and so you get a pretty good concentration response curve. It's not quite as potent. We thought it would have sub-micromolar potency. It might be because some of these, they might have difficulty getting to some of these sense cells, these chordotonal organs that are buried, near the body wall underneath the musculature. We've combined flonicamid and its metabolite with permethrin. Again, here's our permethrin dose response curves. This is permethrin by itself to Anopheles gambiae, the susceptible G3 strain that's shown over here. These are the two response lines that we get with permethrin plus flonicamid in the red circles and permethrin plus the metabolite in the blue squares. They overlap pretty much perfectly. When we look at the curves of flonicamid and 1369 with what we would expect to be additive, so these dashed lines, okay, in the orange and blue are theoretical additive values of what we would expect to see, and we see greater than additive toxicity. So there is synergism if we look at the LD50 values, okay, permethrin alone, right, co-applied with the metabolite, it goes from 0.2 to 0.1. The additive effect would be 0.155. The same kind of analysis co-applied with flonicamid, 0.098, and then the additive effect that we would get would be 0.137. Both of these effects are statistically significant. If we compare the co-applied to the theoretical additive, it's significant both for 1369, the amide metabolite, and for the parent flonicamid itself. So it's not a high level of synergism, but it is synergism as it exists. And I'd like to wrap up the data portion of my presentation by talking about some more recent data that we've collected over the past few months, looking at the synergism of permethrin by certain kinds of essential oils. There have been reports that essential oils are able to synergize the toxicity of pyrethroids, and this is performed at 10 micrograms per mosquito. We're also comparing it with piperno butoxide at 0.5 micrograms. This is in Aedes aegypti adult females. And this is just a combination experiment. We got two different doses of permethrin, the LD20 at 0.04 and the LD50 at about 0.09. I've drawn these lines here. Here's permethrin by itself at 0.04. It's a calculated LD20. We got about 7% toxicity in these particular experiments. And then for comparison, I've just drawn that line across this large bar graph. At 0.09 nanograms per milligram, you can see LD50, permethrin was causing about 40% mortality, pretty close to the LD50 value. If we pre-treat with PBO at 0.5 micrograms, you see we get a statistically significant elevation of both the 0.04 and the 0.09 toxicities that we would see with permethrin. Nothing much in here. In the middle of these was black pepper or camphor, cedarwood, dill, frankincense, helichrysum, lemon, or myrtle. All of these essential oils really had no statistically significant effect. And you can see at 0.09, they're all hovering right around the LD50 value here. Sandalwood and thyme were pretty active. Both of these were able to synergize permethrin at both doses. Then we reach another desert of activity. There's not much out here with sage, marjoram, eucalyptus, bergamot, fennel, or tea tree. But then cucumber seed oil was extremely effective in a sample that we purchased commercially. And so we thought we would investigate these further. And so this is the permethrin at the LD50 dose, or at this applied dose, I should say. It gave us 60% knockdown in 1 hour and 24-hour mortality of about 7%. When we add cucumber seed oil at 0.15 micrograms, the knockdown stays about the same. The mortality goes up at 24 hours, but this is not statistically significant because of the variability. Sandalwood combined with permethrin, 0.052 and 3.6, again, 50% knockdown, 17% mortality. And with thyme oil, 0.45 micrograms, we get a little bit more knockdown, but the mortality doesn't change much. So we really don't see much synergism at these combined doses, at least in a paired kind of comparisons. We have run additional experiments. Now we're looking at the compounds by themselves. We need to know a little bit more about their dose dependent effects on two different strains. We have an Orlando strain, which is our susceptible strain of Aedes aegypti, and a Puerto Rico strain. And here are the LD50 doses with their confidence limits, and it has about 100-fold resistance, this Puerto Rico strain. Cucumber seed oil is rather amazing. amazingly active in our hands. As we studied it, 0.31 micrograms is a really pretty good toxicity. And surprisingly too, when we looked at sandalwood and thyme, which will be coming up shortly, we see a fair amount of resistance to Puerto Rico against this oil, 45-fold, which was very surprising. What we expected to get were resistance ratios like this. This is more the rule, because you've got a complex mixture with multiple compounds. You usually don't see a lot of cross resistance in these oils. And so it also takes about 10-fold more sandalwood or thyme oil to reach an LD50 than it did cucumber seed oil. Then we were very interested in finding out, what is it about cucumber seed oil that's so toxic? Well, turns out that when we fractionated it and did GCMS analysis, that the cucumber seed oil sample that we have is actually contaminated with about 0.5% of chlorpyrifos, which of course is a very highly toxic organophosphorus compound. We have talked to the manufacturer of this oil, and they've given us a set of analyses that were run, and there's no chlorpyrifos in the analysis. So maybe they didn't see it because it's a very small amount, but it seems to be there in our sample. It could be, sampling bias, or we just got a bad sample. I'm not sure yet. But this amount of chlorpyrifos is just about enough, we think, to explain the toxicity we see in the susceptible. But we don't know exactly what's going on in the resistant strain even now, because the resistance ratio for chlorpyrifos in that Puerto Rico strain of 80s Aegypti is only about 5, whereas recall in the whole oil comparison between susceptible and resistant, we get a resistance ratio of about 45. So there's something interesting going on there. We don't know what it is yet. Okay, then, in conclusion, I think the Synergist data is useful, particularly when we use it in the lab for pinning down resistance mechanisms, but it needs to be interpreted with some caution. There's a lot of rather a casual interpretation of Synergist data. Yes, no, P450, carboxylesterase, and GSTs, and we need to think more about maybe its effect on penetration as well. We find this 2S65, 465 compound was an excellent pyrethroid synergist. The behavioral effects supported joint action on nerve. Remember, we had the twitching legs that fell off of the mosquitoes, and Xiao is poised now to study the effects on the nerve membrane itself. Flonicamid metabolite had some potent and selective effects on Anopheles gambiae. We also saw synergism with that and the parent flonicamid. It was rather modest. We see the same sort of modest effects for essential oils. And I think one thing for sure is that anybody working on essential oils needs to check the composition, and even not necessarily rely on analyses that have been done by the manufacturer. In conclusion, then, I would like to acknowledge support for the past few years and efforts from people in my lab. The Deployed Warfighter Protection Research Program has funded Xiao Zheng, Aaron Gross, Jania Taylor-Wells, who now works for Oxitec, and Fabian Damar. Aaron is now at Virginia Tech, and he's there with Paul Carlier, the synthetic chemist I work with, and Joseph Clemens. Some of the essential oil work was funded by our Southeastern Regional Center for Excellence from the CDC. June Tack is the postdoc who ran those experiments with another grad student, Arjun Kotka. We got mosquitoes from Dan Klein, Aedes aegypti from the USDA, and we thank him for it. Same with Paul Howell and the Anopheles gambiae. And finally, I'd like to thank Bao-Nan Sun, a previous postdoc on our vector control grant that we had from the Global Challenges in Global Health, who did all of the patch clamp studies that I've shown you this afternoon. Thank you very much.